Welcome to this overview of male reproductive physiology, where we'll explore structure, function, and regulation across the lifespan. This presentation will move from testicular development and spermatogenesis to hormonal control, accessory glands, and practical factors like thermoregulation. The icons on screen hint at the topics ahead: hormonal control, sperm production, supporting glands, and the pathways that connect them all. This timeline shows how testes form before birth, descend by delivery, then enter a quiet prepubertal phase with low HPG activity. At puberty, the hypothalamic pituitary gonadal axis reactivates, driving spermatogenesis and adult endocrine function. And later in life, a gradual senescence reduces, but does not eliminate function. Think of it as a staged activation and slow decline spanning decades. Spermatogenesis takes place inside seminiferous tubules over roughly 70 days, starting from spermatogonial stem cells and progressing through meiosis to mature spermatozoa. Sertoli cells provide structural support, nutrients, and a blood-testis barrier while coordinating development, ensuring millions of sperm are produced daily and maintaining lifelong fertility potential, as illustrated in the diagram. Hormonal control of sperm production relies on a two-hormone partnership. LH stimulates interstitial Leydig cells to make testosterone, while FSH acts on Sertoli cells to support maturation. Testosterone diffuses into tubules, and together with Sertoli-derived proteins, creates locally high androgen levels, essential for normal sperm output, forming an integrated system that links endocrine signals to cellular maturation. The hypothalamus controls reproduction through pulsatile GnRH release, sending hourly signals to the pituitary to regulate LH and FSH secretion. Those gonadotropins then drive Leydig and Sertoli cell function, ultimately determining testosterone levels, spermatogenesis, and libido. Pulsatility is key, and factors like stress, nutrition, and medications can disrupt the rhythm and impair downstream reproductive outcomes. Hormone balance is maintained by a precise negative feedback loop, where testosterone and inhibin suppress hypothalamic GnRH and pituitary FSH-LH to stabilize circulating levels. Introducing external androgens bypasses this control, lowering intratesticular testosterone and rapidly reducing sperm count and testicular volume by exploiting the same feedback mechanisms, a reversible but impactful suppression of production. After leaving the testis, sperm enter the epididymis immotile and infertile, then transit the corpus where they acquire motility and surface modifications, and finally reach the cauda for storage and ejaculation readiness. Region-specific secretions, absorption, and acidification over about 10 days fine-tune the sperm surface and function, while temperature and inflammatory insults can sensitize or disrupt this maturation process. Ejaculation is a coordinated sequence driven by sympathetic, somatic, and autonomic inputs. Sperm move from the epididymis into the vas deferens and through seminal vesicles and prostate, while rhythmic pelvic floor contractions under somatic control propel the ejaculate. A coordinated bladder neck closure prevents retrograde flow, ensuring efficient expulsion of sperm and seminal fluids during climax. Seminal vesicles contribute roughly 60% of ejaculate volume, producing fructose to fuel sperm glycolysis, prostaglandins that influence female tract motility and immunity, and clotting proteins that stabilize the ejaculate after deposition. Their secretions are regulated by testosterone and sympathetic innervation, and together they play a key role in supporting sperm energy needs and early interactions in the female reproductive tract. Accessory glands create an optimal microenvironment for sperm survival and transport. The prostate secretes enzymes to liquefy semen, citrate and zinc to buffer vaginal pH, and antimicrobial factors to protect sperm, while bulbourethral glands provide pre-ejaculatory mucus and neutralizing fluids to lubricate and clear the urethra. These combined secretions facilitate sperm ascent toward the oocyte. Maintaining a lower testicular temperature depends on scrotal reflexes and countercurrent heat exchange in the spermatic cord, keeping intratesticular temperature about 2 degrees below core body temperature. When regulation fails, due to varicocele, undescended testes, or prolonged heat exposure, spermatogenic efficiency drops and oxidative DNA damage increases, highlighting the importance of thermoregulation for fertility. With aging, Leydig cell numbers and steroidogenic capacity decline gradually, producing about a 1% per year drop in serum testosterone after age 30 and resulting hormonal feedback changes with higher LH. Clinically, this links to reduced muscle mass, libido, and bone density, though sperm output is relatively preserved into later decades, reflecting a slow shift rather than abrupt loss of function.